Hi everyone, welcome to Unit 3, Chapter 4, our last lecture of the semester. As always, make sure that you click on this link to print out the guided notes, which you can turn in for extra credit after the exam. Now, the Unit 3 exam is going to be coming up next week, next Friday, but after that, you have your cumulative final. Okay, and the cumulative final will cover Unit 1, Unit 2, and Unit 3, but it's open notes. So make sure that you keep all of these notes and have them with you. You will not be allowed to use a phone or other devices, but you will be allowed to use any form of paper notes. So these end up being very helpful at the end of the semester for your final. Now we're going to talk about different ways that genes are regulated and biotechnology. So looking at bacteria, bacteria have this one gene, one polypeptide. They don't have a whole lot of extra DNA like humans do, where we have introns and extrons. The bacteria just have one gene, one polypeptide, no introns, no exons. That, however, is just kind of an introduction to the idea of an operon. Humans also have operons. An operon is a group of genes. For instance, if I have an operon and I want to make a smiley face, okay, I'm going to have one gene that creates the circle, one gene that creates the eyes, and one gene that creates the mouth. I need all three genes to make a smiley face. I need all three of them. I don't need just circles and eyes, and I don't need just eyes and mouths. I need all three at the same time. So in order to streamline this process, they're all going to be connected to the same operon. Okay, an operon is made up of three parts. The main part of an operon is going to be each individual gene, and there may be many genes or only a few. The examples that we're going to look at are between 3 and 5. Okay, the other two pieces of this puzzle are the promoter. And if you remember from last week, the promoter is usually made up of a series of repeating TAs, so thymine and adenosine. We call this the TATA box, and this is a really attractive area. Since we have that repeating pattern, it's easily recognizable. And guess who recognizes it? It's going to be polymerase. Okay, I'm just going to put a P for polymerase. The next section is going to be the operator. Now, this is a section of DNA where a repressor may or may not bind. If the repressor is bound, it will turn the gene off. If the repressor is not bound, it will turn the gene on. This polymerase is always going to be pushing forward, but if it runs into a repressor which is bound to this area, it will cause it to fall off. If it does not run into a repressor, it will continue forward and it will transcribe each of these genes inside of this operon. So in that way, the gene is regulated. It can either be turned on with a repressor or turned off with a repressor at this operator location. So we have the three parts, the promoter and the operator, and then the three genes. Now, these operons, this operator area, it can either be turned on or off, meaning it is either always on or always off. If it is always on, that means that polymerase will continue to make copies of these genes indefinitely, unless it is turned off. That means it is repressible. If it is always off, that means it is never translating these genes. These genes are never going to be turned into proteins unless we turn it on, and that means it is inducible. Okay, so we have two different types of operons, a repressible operon or an inducible operon, and they are both going to be made up of a promoter and operator and then the collection of genes that we're trying to copy. Let's look at an example. For example, we have something called the TRP or the trip operon. Now, the TRP operon is an operon that creates a protein called tryptophan. And tryptophan is made up of 5 different subproteins. So we want to translate all of these proteins all at once. So we're going to transcribe them all at once with the TRP operon. Now, the TRP operon is repressible, meaning it's always on. In the normal state, the RNA polymerase is always going to be moving forward no matter what. But here's the thing, tryptophan, our end result here, perfectly fits into our repressor, okay? And we've already learned about proteins, that proteins can sometimes have an active area, and when something fits into their active area, they become activated. This repressor, when tryptophan binds to it, will cause it to bind the operator and turn off the operon, or turn off every single one of these genes. It's interesting because as we create more tryptophan, it will cause less and less tryptophan to be translated. So this is going to end up being a feedback loop, meaning if this is our level of tryptophan and this is time, we have a threshold. And as we create more and more tryptophan, if we cross that threshold, it will turn off the production of tryptophan and the levels will drop below the threshold, which will then tell it to turn back on. And as we cross and create more, it will tell it to turn back off. This is very similar to your thermostat at home. When the temperatures become too hot, the fan will kick on, which will lower the temperature. If the temperature becomes too cool, it will turn off the fan, which will raise the temperature. And we end up holding steady in this threshold here, in this little area of not too much and not too little, because we always want about the same amount of tryptophan in the cell. So this is a repressible gene that is always on, and the end result, the tryptophan, actually binds to the repressor, which will turn the gene off. And all that the repressor does is bind to the operator area of the DNA, which prevents DNA polymerase or RNA polymerase from transcribing the gene. Let's look at a different example. This is an example of an inducible gene, meaning it's always off. This example is called lac operon. It creates lactase. Lactase is important for breaking down the lactose sugar, which is a milk sugar. Now, some people actually don't have this gene. If they do not have this gene, they will be lactose intolerant, meaning their body cannot break down the lactose sugar. And so the sugar will stay in their colon. And as we learned earlier, if we have a whole lot of sugar in one area and not in the other, then water will be drawn to that area, meaning water will be drawn into the colon, causing diarrhea. So very uncomfortable for those who are lactose intolerant and do lack this gene. But those who have this gene, the repressor will always be bound to the operator. It is just naturally going to be bound to the operator all the time, meaning RNA polymerase can't really move forward ever. It's going to bind to the TATA box, okay? And then as it moves forward, because it always is trying to move forward, it will bump into this repressor and break apart, and then it will bind to the TATA box again, and it will just be in this fatal loop, unable to produce any lactase. Now, lactase is only needed if you have lactose in your digestive system. So it is signaled by having lactose or allolactose, a milk sugar, binds to the repressor, which causes the repressor to release from the operator, allowing RNA polymerase to copy this operon, which will then create the lactase, which will break apart the lactose, which will remove it from the repressor, which means the repressor will bind to the operator again. So this is a perfect loop that will only produce lactase when there is lactose or the sugar available to be broken down. Once there is no more lactose available to be broken down, the repressor will bind to the operator again, preventing further transcription of this gene. So this is an example of an inducible gene that is always off unless you turn it on. So let's look here. This is another type of feedback loop. For instance, if you eat a large meal, you have lots of glucose in your blood, or your blood sugar raises. This sends a signal to your pancreas to create insulin. Insulin is processed, and it is a hormone that causes your blood sugar to be transformed into glucagon or absorbed by your cells. If you don't have enough blood sugar, it sends a signal to release the glucagon from your liver back into your bloodstream, thus raising your blood. So this is another example where we will see it raise and lower within that threshold to prevent the blood sugar from becoming too high or too low. Now, this process does not happen in those who are diabetic because the pancreas has lost its ability to produce insulin. So you will see larger spikes and lower lows because they do not regulate insulin normally. They don't regulate blood sugar normally. They cannot produce insulin and have to take artificial insulin. So depending on the type of diabetes will depend on the type of medication, but the artificial insulin will be given maybe before a large meal so that they don't go through these extremes. Looking at this example, it says which type of operon is always on unless it's turned off. So our example is the trip operon. The trip operon is going to always be on unless it is repressed. So this is an example of a repressible gene or a repressible operon. What type of operon is always off unless there is a start signal? Our example is the lac operon. Well, this would be an example of an inducible gene. It's always off unless we induce it to start. Some other ways that genes can be regulated. So after transcription takes place, all of those introns have been cut out of the RNA, which means we have all of these fragments of microRNA. And those fragments can actually play a crucial role in the translation process, mainly by blocking translation. So as these little mini pieces of RNA are produced, the microRNA may get stuck to the messenger RNA and prevent it from becoming translated. So translation would be blocked because the ribosome cannot continue working on this messenger RNA if it's bound to another piece of microRNA. So it will block transcription and cause the mRNA to be recycled. Another thing that can happen is something called a hairpin loop, where the RNA will actually loop around and bind to itself at different levels, and this is also unable to be translated. So this kind of blocking of a gene or gene expression is going to happen after transcription and before translation. Another form of regulating gene expression is through acetylation and methylation. This actually doesn't have anything to do with RNA, but it has everything to do with DNA. Now, naturally, DNA is wound around these things called histones. It's a complex of 4 different proteins that wrap up the DNA kind of like beads. Now, depending on how tightly that DNA is wrapped, the RNA polymerase may or may not be able to reach the DNA. Remember, RNA polymerase has to create a transcription bubble. And if it's very tightly wrapped around the histone, we will not be able to make a transcription bubble to be able to start the transcription or gene expression process. So if we want to turn the genes on, we want to loosen the grip around this histone. And if we add an acetyl group, such as this one, the DNA is not very attracted to acetic acid or acetylation. And so the DNA will kind of get loose or unwound from that histone, meaning that we are turning on transcription because we are allowing a transcription bubble to be made in this area. If we want to turn off the gene, we want to tighten that grip around the histone. And if we tighten the grip around the histone, we want to make the histone more attracted to the DNA. And the histone is really attracted to methyl groups. So DNA methylation can cause the DNA to bind or hold on to the histone very tightly, thus turning the gene expression off. Now, these are all different methods of turning genes on and off. And none of it actually has to do with the code that the base pairs are coding for. This all is something called epigenetics. And epigenetics can actually be inherited. One of the ways that we can track this is by following twin studies. So, for example, these two gentlemen, they are famous twins. They are NASA scientists, and one of them spent a year in the International Space Station. And when he returned, there were some marked differences between him and his identical twin brother. Now, identical twins happen when a zygote is formed. So we go through meiosis, and we end up with a sperm or an egg. So in the meiosis process, we end up with 4 gametes, and they have half the amount of DNA. So let's just randomly choose one of these guys, and we will add that to another ovum. Now, both of these only have half the amount of DNA, so we have to add them together to create a complete zygote that has the correct amount of DNA. Now, this zygote is a viable zygote. It could possibly become a full human being if it implants and has a successful pregnancy. But what happens with identical twins is that before this zygote attaches to the uterine lining, it may fracture or split in half, leading to 2 identical clones. And then they will continue down to the uterus, and they will implant it with separate placenta and separate umbilical cords. Now, depending on when this happens, sometimes they can separate later and end up with the same placenta or even the same umbilical cord. If it separates too late, then we can end up with conjoined twins, which is not the best outcome for the offspring. But if it happens early enough, we end up with identical twins. And identical twin studies are very important because, like I said, if I did a genetic test at this stage, these 2 zygotes would be genetically identical, genetic clones. But as we can see with these young ladies, one of these sisters has spent a lot more time in the sun than the other. So Sister B may have had a career like gardening where she spent a lot of time outdoors, or maybe a mail carrier, so she spent a lot of time outdoors. So of these 2 sisters, if we did a DNA test right now, they would mostly be similar, but there may be some marked differences. Mainly the instance or the susceptibility to skin cancer. So I would assume that Sister B, who has spent a lot more time in the sun, may be more susceptible to skin cancer. Now, if both of these sisters had a daughter, then I may also assume that the daughter of Sister B has a higher chance of getting skin cancer, even if daughter B does not spend time in the sun. This is due to epigenetic inheritance. It doesn't actually have to do with the DNA base pairs that are inherited, but more of the patterns of gene expression. Let's go over a different example. Let's look at these 2 mice. This mouse is a mother mouse, and she was kept in an enriched environment. This mouse, however, was not kept in an enriched environment. In fact, they stressed her out a lot. This is a real experiment that really happened. I don't know if this would pass the ethics review board today, but we did learn a lot of important information from this mouse study. So we stressed this mouse out, which means that this mouse, when she had pups, was a very affectionate mother. And this is a measurable trait. They measured how much time she spent with her offspring. This mother, however, after being stressed out, now she's put into a normal environment, even though she's not in this stressful environment anymore. Now she's in a normal, healthy, enriched environment, does not spend as much time with her pups as the first one did. So what do we see as an outcome? Well, if I put this pup into a slightly stressful situation and then remove it from that stressful situation, it recovers very quickly from the stress. It has a normal stress response. But these pups, if I put them in a stressful situation and remove them from the stressful situation, they still behave as if they are in the stressful situation. So they do not recover from that stressor very well. They don't respond very well to stress. So let's hypothesize. If this mouse has a pup, do you think that she is going to be an affectionate mother or a detached mother? Well, you may have guessed that she ends up being a rather affectionate mother in a normal environment. And her pups also become very well adjusted and respond normally to stress. What about this mouse? Do you think that this mouse is going to be an affectionate mother or a detached mother? You may have guessed that she is a detached mother. Now, this mouse was never in the stressful environment that this mouse was in. In fact, this mouse was only in that stressful environment prior to having pups for maybe a week. This pup never experienced that kind of stressor, but still became a detached mother. And her pups do not respond well to stress either. Any stressful stimuli and these mice somewhat shut down. So we have 3 generations from grandmother to granddaughter mouse. We have 3 generations of maladjusted pups who don't respond well to stress. And this is due to their epigenetic inheritance. They did inherit the same genes, but they did not inherit the same behaviors. One of the reasons we think this happens is through epigenetic inheritance, traits that are not passed down from the nucleotide sequence, but by the modifications of the actual chromosomes. And we see this in subsequent generations. So, for instance, if this is me right now, okay, I had all of my ovaries and all of the eggs that I'm ever going to produce while I was a fetus inside of my mother when she was pregnant. My mother was inside of my grandmother and had all of her eggs at the time that she was pregnant. So that means that the egg that was selected to produce me actually already existed while my grandmother was pregnant in 1961. Okay, so back from 1961, the egg that was selected to produce me was already in existence. That's because of the process that we talked about earlier, meiosis. Only men, when they undergo meiosis, they can produce new sperm cells throughout their lives. Women, however, they go through all the meiosis that they're ever going to go through while they're a fetus. So all the eggs that she is ever going to produce already exist, at least partially, before she is even born. So that means the actions of my grandmother in the 1960s can affect me today. And one of the things that was really common in the 1950s and 60s was smoking. It really wasn't until later that we realized that smoking was bad and a carcinogen and was, definitely discouraged during pregnancy, but really discouraged anytime. And one of the effects, long-term effects of smoking was childhood obesity. So if a woman who was of normal body mass smoked, her child had a higher likelihood of developing obesity, and even her child has a higher likelihood of developing obesity because the egg that was selected for fertilization already existed in the grandmother's pregnancy. Back for me, it was 1961 because my mother was born in 1961. So for you, it might be a little bit different timeline, and so your grandmother may have not been a smoker. So it just depends. We're just using this as an example. But this is a type of epigenetic inheritance where the actions of previous generations can actually, and the experiences of previous generations can actually affect the up to 3 generations. I think that's pretty interesting. All right, let's talk about other things that can change, like mutations. There are several things that can actually cause mutations, such as radiation. One of the most common forms of radiation is a sunburn. So a sunburn can cause radiation. We also have something called, chemicals called carcinogens. Carcinogens such as cigarettes and some cleaning products can cause mutations, which is why it's always a good idea to wear gloves when cleaning and don't smoke. I'm not sure why it keeps doing that. All right, but also infectious agents can also cause mutations, such as viruses. For instance, the HPV virus, the human papillomavirus, is a very common virus that is sexually transmitted. Now, it doesn't have a really big effect when you become infected. It may cause a few genital warts, but otherwise not really noticeable. So many people pass it on to their sexual partners without realizing it. Now, HPV, like I said, is generally pretty harmless, but there's a very small chance that that viral infection can later lead to cervical cancer in women. So that's why HPV is commonly screened for annually during a woman's annual checkup. Now, what kinds of mutations occur? Well, there are different types of mutations. The most common type of mutation is a point mutation. Now, DNA polymerase and RNA polymerase hardly ever make mistakes, but sometimes mistakes occur, and that can lead to a point mutation. Otherwise, these mistakes are usually due to mutagens. So, one type is a point mutation where I might just accidentally substitute one of these bases for another. So, replace this C with a G. And as we learned last week, these are read in triplet. So methionine is always going to be the start codon, and then we have something over here, which is the stop codon. If this happens on the third location of the base pair, it probably, or the third location of the codon, it probably won't make a big difference. If it's on the second or the first location, it may change the amino acid sequence. So this could have a result, like it could be visible or it could be silent. This is called a silent mutation when it has no effect at all. Also, if the mutation happens after the stop point or before the start point, it will be silent and not have any effect at all. Now, let's say that it did change one single amino acid. So instead of glycine, maybe it created leucine. All in all, this protein might still be functional. It may not be perfect, but it may be functional, and we call that a missense mutation. We change one amino acid, but overall the whole protein is not affected. There are some bigger kinds of changes though. For instance, an insertion or a deletion. If we deleted or added an extra base pair, it will cause something called a frame shift, which means every single amino acid after that will change. So we have shifted all of the frames to one side. This is going to be a nonsense mutation. It will halt the translation entirely. The protein will not be functional and your body will start over. So these kinds of issues will completely stop the function of that protein. There is a short video which I find really informative if you want to watch that with sound by viewing the PowerPoint. We will not watch it because it is silent on these presentations. So let's look at this example here. We have a normal sequence of C, T, A, G, and remember we read in triplets, so as long as there's not any changes with this single triplet, it will be a-okay. Now, it looks like we inserted a base right here, which will cause everything after it to shift. So this is not only an insertion, but it is also a nonsense mutation, and it is also a frame shift mutation. Then it looks like we deleted one, which also changed all of our base pairs, and it frame shift this direction. So it is also a nonsense, it is also a frame shift because it's a deletion. If there's a duplication, it's kind of like an insertion. It will also cause a frame shift. If there is an inversion, this may or may not cause a big problem. Let's look at this example. So in this example, the whole sentence represents the protein. Each word represents an amino acid, and each letter represents a base pair. So my entire protein originally says the fat cat ate the rat. The fat cat ate the rat. That's what the sentence is supposed to say. Let's look at this first sentence. This first sentence says the fat tat ate the rat. So right away I notice that there is a point substitution. So this would be a point substitution right there, point substitution. But it is going to cause a lot of problems because this sentence no longer makes sense. The fat tat ate the rat does not work. So this protein will not be functional. Let's look at the next one. It says the fat cat ate the bat. Here's the same thing. It is also a point mutation, but this sentence makes sense. It actually works. It doesn't work as well as the original, the fat cat ate the rat, but I think a cat could possibly eat a bat if it had a bat available to it. So we're going to call this a missense mutation because it changed one amino acid, but overall the protein still works. Now let's look at this one, the fat at. Well, the obvious thing is that we had a deletion. So we deleted something, which also caused all the frame shift. So everything frame shifted this direction, which ended up causing complete failure of the protein. The last one, the fat cat, it's a mess. It's awful. And that's because this was inserted. There was an insertion here, which also caused a frame shift. So it's both a frame shift and an insertion, which means that this protein is completely non-functional. So the only mutation that really wasn't detrimental was this one, and that just happened to be because this amino acid works. So these mutations can have a varying degree of effect on the end result. Let's talk about another kind of malfunction that can happen. Now, during meiosis, we talked about how half of the DNA is going to go to one cell and the other half of the DNA is going to go to the other cell. And this is controlled by the spindle fibers. Well, if there is a problem with the spindle fiber, then we might end up with double the DNA on one side and half the DNA on the other side. And this generally is, controlled to just one chromosome. So what we'll end up with is gametes. For instance, if these are all sperms, we will end up with gametes that have an extra chromosome right here and gametes that are missing a chromosome here. So for instance, if this gamete was chosen for a fertilization event, it may have 24 chromosomes, and it will bind to an egg that has 23. Okay, that's a 2, not a 1. It looks kind of like a 1. And for instance, this zygote would have something called polyploidy, meaning it has too much DNA, too much. And so you end up with something called trisomy, where you end up with one chromosome that has too much. Now, if I selected one of these for my sperm, instead of 24, it would have 22. And if that joined with a normal egg, then that zygote, instead of having 46, this one would have 47. This one would have 45 chromosomes, which is an aneuploidy, where we have a missing chromosome. So these are both types of non-disjunction. Non-disjunction just means that we didn't separate during meiosis. Let's look at some examples of that. For instance, here I can see that I have trisomy 18. I have an extra chromosome 18. This is called trisomy 18, or a form of polyploidy. It has too many. It's not an aneuploid, it's a polyploid. But how do we get this information? Well, this is called a karyotype. A karyotype is just a way to organize visually the chromosomes. This really doesn't give you a lot of information about what's actually going on inside of the genetics. This is just a generalized picture, but it's really interesting how we get these images. So all cells go through mitosis, and during mitosis, during prophase of mitosis, the chromatin kind of condenses into chromosomes. We want to stop it at this process. So what we do is we take a sample of cells, maybe a cheek swab, and we look at the cells under a microscope, and we're going to chemically induce them to go into prophase of mitosis. And then during prophase, one thing that happens is these spindle fibers start forming. Well, next what we're going to do is we are going to disrupt those spindle fibers. So the cell has distinct chromosomes, but it will never ever divide because the spindle fibers cannot attach to the chromosomes. So it kind of stalls right here in between prophase and metaphase. And this is because of the chemical treatment. Now, this is the perfect opportunity to take a picture. This is what that picture looks like. Then you will have to painstakingly cut and paste each and every one of these chromosomes and compare them in size and line them up according to biggest to smallest, and then the sex chromosomes at the end. This is the 23rd pair. We do have computer programs that make this a lot easier, but you and I have to do it by hand. If you would like an example of that, you can click on this link right here. That's a weird looking arrow, but that's my arrow. If you click on this link, you will go to a karyotyping activity, but we will also be doing this karyotyping activity in lab. So let's look at some karyotypes. If I look at this karyotype, everything appears normal. This is a female because it has 2 X chromosomes, but if I look right here, we have something called trisomy 21. So this is a polyploid trisomy 21, also known as Down syndrome. Down syndrome can cause cognitive delay. It can cause small, low-set ears, a very round, moon-shaped face, and it can also cause large, flat palms with one large palm crease, and some other defects like a higher incidence of obesity and a higher incidence of heart disease. But a person with Down syndrome can grow to adulthood and live a full, fulfilling life. Now, it does vary in the severity of the disorder. Some persons with Down syndrome will need adult care their entire lives, and some persons with Down syndrome are able to live happy, healthy, independent lives on their own. Everything just kind of depends on the type of Down syndrome and how severe it is. Another example is trisomy 18, which is known as Edwards syndrome. Edwards syndrome is much more dangerous to the offspring. In fact, they generally do not live more than a few months if they have this disorder. Some marked differences is this prominent skull bump at the end, mal-shaped ears, and highly tonic muscles, meaning their muscles are clenched. So they have this fist clench, and their muscles tend to be clenched, which leads to this like plate-shaped chest because the chest muscles are also clenched. And this unfortunately will cause death very quickly. If we look, this is a baby girl because she has 2 X chromosomes and does not have a Y chromosome. This is also a form of polyploidy, trisomy 18. Let's look at this one. This is aneuploidy, where we have a partial deletion of chromosome 5, which means we're only missing part of it. And this leads to something called cru de cat syndrome, which literally translates to cat's cry, because one of the most marked side effects is a deformity of the vocal cords, which actually cause the cries of the baby to sound very similar to a cat crying. If you would like to know what that sounds like, you can look it up on YouTube. It's indistinguishable from a cat. One of the other symptoms is blood pooling in the hands and feet, or like swollen extremities. You do have low set ears, a swollen head, and I'm not sure of the life expectancy of a baby with cru de cat syndrome, but I do believe that they have a shorter life expectancy. So this is a form of aneuploidy. Let's look at this person. All of their chromosomes appear very normal until we get to their sex chromosomes, and they are female because they have 2 X chromosomes, but they're also male because they have a Y chromosome. Okay, so this person will most likely present male through childhood, but then during puberty will start to develop more secondary female characteristics, such as breast development in 30% of the cases, or very female patterned pubic hair and lack of a beard. They will appear more feminine, but persons with Klinefelter syndrome may live a normal, happy life without ever knowing that they have Klinefelter syndrome. They do have a higher instance of osteoporosis because they do have more estrogen than most males do. This can also lead to a form of intersex where the male genitalia is not completely formed at birth, and it may be hard to tell through the genitals whether it's a male or female. But most likely it's going to present as a male with maybe reduced testes. This person will most likely find out that they have this syndrome at puberty. Here is a different example. This person is definitely male. They have 1 X and 2 Y. Now, I do believe that this person is going to appear male upon birth and throughout childhood, but after puberty may have like a larger but less muscular body, like maybe appears a little with less muscular definition than a normal male. They will have longer legs. They may or may not produce a little bit of breast tissue. Very similar to Klinefelter syndrome, but the main difference is that they will present male. They will entirely present male, with a couple of differences after puberty. Now, there is some debate about Jacob's syndrome even causing symptoms, because the way that we found out about Jacob's syndrome, or the way that we studied Jacob's syndrome, was through prison studies, and there was this instance where we saw more Jacob's syndrome in prison than in the public, which led researchers to go down this rabbit hole of believing that maybe Jacob's syndrome causes people to become more aggressive because they have that extra Y chromosome. They must have extra testosterone and be extra aggressive and more susceptible to committing crimes. That I don't necessarily believe. What I think that study says is that we tested for Jacob's syndrome more commonly in prison situations because of that study. There's really no solid evidence that any of those things are true. In fact, a male could have Jacob's syndrome and never know it his entire life. Now, this young lady is a female. She does present female, but she only received one X chromosome, and she did not receive the second chromosome from her other parent. This is called Turner syndrome, and it is a form of aneuploidy or deletion where they are missing one of the sex chromosomes. This little girl will present female at birth, and she will have several telltale signs of Turner syndrome, mainly a thickened neck. Her neck will become thickened, and she is susceptible to getting these brown spots on her skin, which don't normally cause any problems. But the main signs of Turner syndrome will happen around puberty, where she will not go through puberty. She will not produce breasts, or she will not develop breasts. She will not produce any estrogen in her ovaries. They will not be. She will not undergo menstruation. So she will be infertile. I believe that a patient with Turner syndrome can undergo hormone therapy, but it's not going to fix her reproductive organs. So she will still be infertile because she is missing this X chromosome. But female presenting child. All right, we talked about some things that can go wrong. Let's talk about some ways that we can use this information. So one of the really, really cool things that we have in biotechnology is because of forensic science, or is used for forensic science. So let's say that we take a sample of DNA. This sample is going to be really small, especially in the cases of forensic science, because if I'm taking a cheek swab, I can get in there and get as much cheek matter as I want to. But if I'm at a crime scene, I am limited to the drop of blood or the little bit of DNA that is available. And so we need a process to be able to copy that DNA. And the process that we use is called polymerase chain reaction, which is a really, really cool process. What we do is we cycle this DNA inside of a PCR machine. Now, the only thing that the PCR machine actually does is raise the temperature, lower the temperature, raise the temperature, lower the temperature, raise the temperature, lower the temperature. And each time we raise the temperature and lower the temperature, we are doubling the DNA. So when we raise the temperature and lower the temperature, we call that a cycle, which only lasts like a minute and a half. So each minute that our sample is inside the PCR machine, we will double our DNA sample. So if we start out with 80 fragments of DNA, we're immediately going to get 190, and then we're immediately going to double that, to double that, to double that. And after only a few cycles, after about an hour, we end up with millions of copies of the DNA. Now, our DNA polymerase would not be able to withstand these high heats. This high heat is nearly boiling. So we have to have a special polymerase to work inside of this machine. In fact, the machine was built around the special polymerase called Taq polymerase, TAQ polymerase. Now, Taq polymerase is very thermostable. That's because we took it from a bacteria called Thermus aquaticus. This bacteria is known as a thermophile. Phile meaning love, therm meaning heat. It loves the heat. In fact, we found it in Yellowstone National Park in one of the hot springs. So Taq polymerase allowed us to develop the PCR technology because it can withstand these really high heats and still be functional. Let's look at the specific cycles. The first thing that has to happen at step 0, the first thing that's going to happen is we have to mix our materials. So we're going to need a sample of DNA, and hopefully it's a really good sample, because if we don't have a good sample, then we are making copies and copies and copies of a bad sample, and we're going to get a bad result. So we want a good sample DNA. We also have to add a primer, which allows the Taq polymerase to start working, and the primer is going to bind at the beginning and allow the Taq polymerase to move forward. We also, of course, need Taq polymerase, and Taq polymerase would not be able to add these nucleic base pairs if we didn't have plenty of free base pairs. So we're going to add tons and tons of free nucleotides so that Taq polymerase can do its job. The last thing we're going to add is a liquid buffer solution. Not only does this allow everything to kind of swim together in a nice little soup, but it has lots of liquid in it, which means when we reach these really high, nearly boiling temperatures, it's not going to evaporate as quickly because we have plenty of solution. So after we load our tray, which happens at step 0, we go through step 1. Step 1 is called denaturing. During denaturing, we are raising the temperature to about 95 degrees Celsius, nearly boiling. And this will cause the hydrogen bonds between the two strands of DNA to separate. That's why we call it denaturing. Now, we don't want to denature our enzyme, which is why we use special Taq polymerase. The next step, we lower the temperature to about 60 degrees Celsius, which is called annealing. Now, you might think that the DNA will go back together, but since we put the primer in there, it's actually the primer that's going to grab onto the DNA, and the primer will grab onto the beginning of both strands of DNA. Then we are going to raise the temperature to the ideal working temperature of Taq polymerase. This step is called elongation, and it's about 75 degrees Celsius, which is the optimal temperature of Taq polymerase to start doing its job and to start copying the DNA. So it's going to copy all of the DNA. And remember, it's only able to do that because we have plenty of free nucleotides to work with. Then all we have to do is raise the temperature back to step 1, break apart all of those copies, and then lower the temperature to step 2 and add primers to all of those copies, and then raise it to temperature 3 again so that Taq polymerase can make all the copies. So every single time we raise and lower the temperature, we are multiplying our sample by 2. So you can see that we end up with an exponential increase in the number of sample DNAs that we have, and that's only due to raising and lowering the temperature. Now, human body temperature is about 37 degrees Celsius, so this is much higher than any of the human enzymes could handle, because about the optimal temperature of human enzymes is 37 degrees Celsius. So we can see that this process takes a lot more heat than that. And if you would like to view more about that, you can view this video, but it does not have sound when I'm playing it. When you play it, you can view it with sound. Let's talk about what we do with our DNA sample. So let's say that we copied our DNA and we made tons and tons of copies through the PCR. The next thing that we're going to do is break apart the DNA into specific fragments. So we're going to add a splicer to our sample, and the splicer is particularly going to grab onto the Tata box. This is important because it means that we have reliable, regular sized pieces. Each gene is going to be cut at the same place. So if we have really long genes or really short genes, they're all going to be separated because the slicing will happen, or the splicing will happen at the promoter, at the Tata box. But then we will take our DNA and we will load it into something called gel electrophoresis, which is literally a piece of gel. We have a piece of gel and it has these little cutouts at the top. We're going to load our DNA into these specific cutouts. And then we are going to pump electricity through it. Now, this gel is like a sponge. It's spongy, meaning that the DNA will slip through the holes in the sponge, but at varying speeds depending on how big those fragments are. When we put electricity through it, we are adding negative ions, and the negative will always end up going towards the positive pole. Remember, DNA also has a negative charge, so DNA is going to be pulled towards that positive pole. Now, if those fragments are really big, they're going to be pulled really slowly. If the fragments are really small, they're going to be pulled very quickly. So what we end up with is varying sizes of these bands. And usually these bands are not visible until we view it under a special UV light, because part of this process, that's a light bulb, guys, part of this process is to add a fluorescent dye to it. So when you turn on the UV light, you can see the different bands. Now, usually lane 1 is reserved for what's called a DNA ladder, which is an artificial sample that we know has a specific size of base pairs. So for instance, I know that anything at this level has 2,000 base pairs, and anything at this level has 100 base pairs. And then I can compare my actual samples to my control. We can do something called DNA fingerprinting, which we will do in the lab. If you want to watch this video, you can view an actual gel being made. I'm not going to watch the video because, like I said, it's silent, but you can go to the PowerPoint and view this video with sound. Another thing that can happen is cDNA. We can use the transcribed mRNA to go backwards and produce DNA using reverse transcriptase. This is really helpful because our DNA is really, really long, but remember, when we make copies of it, we pull out the introns and we're left with only the exons. So our RNA is much shorter than our DNA. So if we convert that back into DNA, we can have more streamlined cDNA that we use in a cDNA library, or we can compare it and see what percent of the DNA is actually being translated into proteins. So we can compare the mRNA to the cDNA. Again, if you would like to view this video, I highly suggest it, but the sound won't work, so unless you watch it yourself in the PowerPoint. So do watch the cDNA. There is also this process of artificial chromosomes, or bacterial artificial chromosomes. This is similar to the previous process that we talked about, and we can actually use the cDNA that we make to create fake DNA sequences, or a foreign genome. So these recombinant plasmids, we're going to make a plasmid library or genomic library by creating these cDNA sequences. And then when we insert our cDNA sequence, let's say we isolate the gene that we want, for instance, the gene that produces insulin, that tells your pancreas how to produce insulin. What we can do is insert that gene into a bacteria. And then when that bacteria multiplies, we will end up with many, many, many bacteria that can produce insulin. And this is one of the very first commercial uses of these bacterial artificial chromosomes, because we inserted the gene that produces insulin, and now we have a whole bunch of bacteria that its only job is to produce tons and tons and tons of insulin. So this is a really important way that we use biotechnology to create medicines. If you would like to view this video, it is a very important, useful video with sound. So please go back to the PowerPoint and watch this video with sound, where you can view how the recombinant plasmid is used to create insulin. As always, don't forget to do your chapter laboratory write-up and print it out and use it for the OpenNote lab quiz, as well as your OpenNote lab practicals. Also, don't forget to take your chapter quiz for lecture in Blackboard. There will be a practice quizzes after this PowerPoint. And if you choose to do the chapter homework, you can check your work with the SI leader or emailing me, but you can turn those in after your exam for extra credit. Thank you guys for staying with me this semester and working so hard and making my life so much easier by being wonderful students. And I have really enjoyed having you all semester long. And I look forward to possibly seeing you for biology too. Have a great semester.